Before we go further, let me ask this question. To what extent or what degree of changes in the chromosome or DNA that will lead to the changes in phenotype? Well, in this video, we can find the answer for this question. Let's begin with the definition of mutation. Mutation is a permanent change in the nucleotide sequence or amount, arrangements, or structure of the DNA of an organism that creates genetic diversity. An organism that carrying a gene that has undergone mutation or mutated gene is called mutant that shows new or different phenotypes. There are two types of mutation: spontaneous mutation and induced mutation. Spontaneous mutations occurs naturally without human intervention. It's a random or rare event caused by non-disjunction due to mutagens in the environment. Mutagens such as cosmic ray, radioactive compound, and extreme heat that occurs during mistake in DNA replication or lack of DNA repair. Induced mutations occurs artificially, involve human intervention. For example, it causes by the exposure to the physical mutagens such as X-ray at the hospital and chemical mutagens such as colchicine in the laboratory. It contributes to the evolution that produce more variation. Mutagens is a chemical or physical agent that interact with the DNA and will cause mutation. Two types of mutagen: physical agent, high energy radiation such as ultraviolet, gamma ray, X-ray, and cosmic ray. The second type will be chemical agent such colchicine, ethidium bromide, proflavin, mustard gas, formaldehyde, and base analogues. Now, let's look into the gene mutation. Gene mutation or point mutation is a change in the nucleotide sequence of a single gene of a DNA. Changes of a single nucleotide in gene lead to the changes in mRNA, which lead to the changes of amino acid encoded by a codon and produce different protein. If the change involved in a single base, then it is called point mutation. If the mutation occur in a gamete or cell that become gamete, then it can be inherited into the next generation. There are four types of gene mutation. Number one, base substitution. One or few base pairs in the nucleotide sequence in a gene is replaced. The second type is base insertion. Addition of one or few base pairs in the nucleotide sequence in a gene. The third type is base deletion. Loss of one or few base pair in the nucleotide sequence in a gene. Fourth, base inversion. Two base pairs or more are inverted in nucleotide sequence in a gene. Now we will look into the gene mutation, base substitution that will cause sickle cell anemia as an example of missense mutation. In base substitution, one or few base pairs in the nucleotide sequence in a gene replace, transcribe into different mRNA. The altered mRNA will be translated into different polypeptide with different amino acid sequence. Now, let's take a look into the base substitution that will cause the sickle cell anemia. In the DNA of the mutant, thymine is substituted by adenine, and on the mRNA, adenine is substituted by uracil. Amino acid sequence of the beta globin, glutamic acid, is replaced by valine. In this diagram, we can see that the change of glutamic acid into valine will affect the shape of the beta polypeptide of the hemoglobin that will reduce the ability of hemoglobin to transport oxygen. This also will cause the red blood cell change its shape into the sickle shape. Now is the detail of the base substitution that will cause sickle cell anemia. It will involve the base substitution that lead to missense mutation. Gene mutation will alter or will change the sequence of a nucleotide bases. In sickle cell anemia, the nucleotide bases on DNA, CTC, are replaced by CAC. Or, to be specific, thymine is replaced by adenine. The codon on mRNA, GAG, are replaced by GUG during transcription. The codon that is supposed to code for amino acid glutamic acid is replaced by a codon that will code for valine during translation. The hemoglobin in the red blood cells become crystallized or stiff or become distorted. That will cause the red blood cells to change into sickle shape when the partial pressure of oxygen decreases. This will cause the red blood cells to become easily clump or clog or form a blockage into the blood capillaries. That will make the transport of oxygens become less efficient. Base substitution can cause missense mutation. Changes of codon that code for different amino acid that will produce different polypeptide or different protein. The next effect of the base substitution is nonsense mutation. A point mutation can also change codon for an amino acid into a stop codon that will cause translation to be terminated prematurely. The resulting polypeptide will be shorter or truncated than the polypeptide encoded by the normal gene. Most nonsense mutations lead to non-functional proteins. The third effect of base substitution is called silent mutation. A change in nucleotide pair may transform one codon into another, but still translated into the same amino acid. Now, let's focus on this diagram to see the effect of gene mutation. First, in the normal type of mRNA, it consists of AUG that will code for methionine, AAG that will code for lysine, UUU that will code for phenylalanine, and GGC that will code for glycine, and UAA that will code for stop codon. In base substitution, on the DNA, guanine is substituted by adenine, and on the mRNA, cytosine is replaced by uracil. However, the new codon GGU will still code for the same amino acid. This effect is called silent mutation. Next situation is on the DNA, cytosine is substituted by thymine, and on the mRNA, guanine is substituted into adenine. The new codon AGC is coded for different amino acid, and this effect is called missense mutation. Another example of base substitution is when on the DNA, thymine is substituted by adenine, and on the mRNA, adenine is substituted into uracil. The new codon UAG is coded for stop codon. This will cause the polypeptide will become shorter. This effect is called nonsense mutation. Next, we will look into the effects of base insertion and base deletion. For example, the insertion of adenine into the DNA strand will cause the frameshift mutation that will cause the new codon that produced is UAA that will code for stop codon. This frameshift mutation also will cause nonsense mutation. Another example, when adenine is deleted from the DNA strand, this frameshift mutation will cause changes in extensive amino acid that will lead to the missense mutation. For this example, if the deletion involves a deletion of triplet bases, there will be no frameshift mutation occur, but one amino acid is changed or missing. Same goes when triplet bases is added into the DNA strand. This will not cause a frameshift mutation, but will cause the extra amino acid in the polypeptide. For base inversion, two bases pair or more nucleotide sequence are inverted. Changes in the codons lead to change in the amino acid sequence. Base inversion will cause minor phenotype abnormality, the effect of base insertion and base deletion. Mutation that involve insertion or deletion of one or more base pair in the nucleotide sequence of DNA will cause frameshift mutation. In frameshift mutation, shifting the reading frame by one or two bases cause the gene to be read in the wrong codon, caused by base insertion and base deletion of a number of base pair nucleotide pairs. From the point mutation, gene read in wrong codons or triplet bases in DNA and mRNA. Changes in codons result in the changes of amino acid sequence, produce different protein. The effect usually severe and harmful. For example, Tay-Sachs disease and major thalassemia. For the conclusion, even a change in single nucleotide base pair also can lead to the changes of phenotype or can lead to the genetic diversity.